Hi, I'm Wynn Meyer, and my research falls at the intersection of evolutionary biology and molecular biology, specifically genomics. For as long as I can remember, I've been really fascinated with the amazing array of phenotypic diversity that we see on this planet. And since I became a geneticist, I've been really curious about understanding what the genetic basis is for many of these wide array of traits, some of them adaptive. Now that it's very easy to sequence an organism's genome, we can start to get at these questions about the genetic basis of phenotypic diversity. Conversely, just because we know the genome sequence doesn't mean that we understand what it does. And so by linking specific regions of our genomes with some of these phenotypes, we can also start to understand what different parts of our genomes do. Within this broad framework of questions, my lab is currently investigating the molecular basis of adaptation to different diets across mammals. Here you can see a cartoon of the mammalian family tree with different diets highlighted by these different symbols and colors. One thing that you can see is that mammalian diets vary widely and that certain diets have evolved repeatedly or convergently across different lineages of mammals. This combination of variation and convergence means that we have tools to identify particular genes or sets of genes that have been repeatedly involved in the adaptation to the same diet. So one particular method that we can use to identify these genes is a software that I worked on when I was a postdoctoral researcher. Now we're seeing not just a cartoon of the mammalian family tree, but an actual phylogeny, where the lengths of these branches represent the average number of changes that have occurred in a protein along a particular evolutionary lineage. If we compare this average tree with the tree for a specific gene, here, the branches represent the number of changes that have occurred in a particular gene on that lineage. We can see that there are certain genes within the genome where some lineages have evolved more rapidly than you would expect based on this average, genome-wide average tree. In this particular case, these branches that are labeled in green are mammals that have predominantly plant-based diets. And so we can use this to develop a test statistic where we compare the relative rate of evolution of a gene compared to its expectation in these green foreground species with that of the species that don't have plant-based diets, and use this to identify the genes that tend to co-evolve with plant-based diets. Our lab is busy performing these analyses, and we have already identified some promising candidates. So one of the projects that we are focusing on in our future work is characterizing these exciting genes that we've identified that co-evolve with diet. So for instance, we might be interested in what kinds of changes in protein structure do herbivores have in comparison to carnivores. Another project asks not what kinds of changes in individual genes associate with diet, but what changes in copy number of genes within a particular family are associated with diet. And another project looks at these genes that are associated with diet across mammals and asks, how do they evolve within humans? We already know that some of the strongest signatures of recent human adaptation involve genes that are responsible for our ability to digest certain foods, in this case, milk. You might have inferred from the previous slides that a lot of what we do is computational, and that's true. Here's a picture of the Lehigh High Performance Computing Cluster on which we do many of our analyses with large-scale genomic data. We're also involved in generating new genomic data, and for that, we do work in the molecular lab with DNA and RNA sequencing of new and interesting species. In addition to what we directly do in our lab, we frequently collaborate closely with other labs. In some of those labs, you might have an opportunity to do some follow-up work on these genes and model organisms, such as the budding yeast, or you could go and collect new data through field research. Shown here are some bats with high sugar diets that live in East Asia, where one of my collaborators does field research. Beyond our research, we're very committed to making sure that everyone has access to science. And I have some cool outreach projects in the works to try and communicate the power of genomics to educators and community members nearby. I also have experience with science policy, so if you are interested in how science can inform policy or how to improve policy to support science, I would be glad to talk to you about efforts in these areas. If any of this has sounded interesting to you, please feel free to check out my website and get in touch at the email listed below. Thank you so much.